So to begin, human immunodeficiency virus, or HIV, is part of the Retroviridae family. 120 nanometers enveloped particles with conosyl capsid, linear positive single-stranded RNA genome of about 9,700 pairs, thought to have evolved from the simian immunodeficiency virus, or SIV. It is a distant virus that infects humans. Vaccines are currently being tested for it. It was first recognized by humans in the 1980s. The virus infects and destroys the body's immune cells, which means that as the infection progresses, people lose their ability to fight off the disease. So in turn of making itself stronger, it makes the body's defenses weaker. Acquired immunodeficiency syndrome, or AIDS, refers to the advanced stages of HIV infection when a person's immune system is severely impaired. Because HIV can permanently incorporate its genome into that of a host cell, the infection lasts a lifetime. There's currently no vaccine against HIV, although numerous drugs can keep infection at bay and hinder disease progression. Nearly 38 million people worldwide were infected with HIV by the end of 2019. In part A of this diagram, we witness the HIV virus and the glycoproteins bind to the receptors on the host cell surface. In part B, we witness it trigger the cell membrane to fuse with the virus's envelope, releasing the virus's positive RNA genome and viral proteins, including the reverse transcriptase and integrase, into the cell's cytosol. In part C, the viral reverse transcriptase transcribes the virus's positive RNA genome complementary DNA in a process known as reverse transcription, and since it is positive RNA, it directly happens. In part D, it is that then the dsDNA copy of the virus's genome travels to the nucleus, is inserted into the cell's genome by viral integrase. The viral genome is then transcribed by the host machinery alongside the host genome. In part E, this produces more copies of the virus's positive RNA genome. In part F, as well as more mRNA, or messenger RNA. In part G, the messenger RNA leaves the nucleus and is translated into proteins by ribosomes, into the cytosol and onto endoplasmic reticulum. Part H, HIV glycoproteins are further modified through protein processing in the Golgi network. Part I, then they're transported into the cell membrane. Viral genomes and proteins assemble into the new viruses at the cell surface. In part J, which leave the cell through the process known as budding. And finally, this process surrounds the virus in a piece of cell membrane containing viral proteins, which becomes its envelope. And you must be asking, Sam, how are you so sure everything you say is true? To that, I tell you,
we're not just sure, we're HIV positive.